Good morning, everyone, and welcome to day 2 of the Future of Fieldwork. I have met many of you already, but just in case you haven't met me, my name is Michelle McDonald, and I'm the director of the Library and Museum here at the American Philosophical Society. How many of you were here yesterday? Almost all of you. Okay, so yesterday you met Patrick Spiro when he was doing the introduction, our Chief Executive Officer. I'm just like Pat, except I'm 10 years older, I'm female, and I have gray hair. Other than that, just like Patrick, not really. So my job is to welcome you to the APS this morning. I'm going to give an abbreviated version of the APS history for just, who wasn't here yesterday? Okay, for you, sir. I am going to give an abbreviated version of the APS history. For the others, it's a bit of a refresher. But first, I'm going to start with our land acknowledgement, which we use at our public programs to acknowledge where we are and what came before us. So the American Philosophical Society resides in Lenape-Hoking, the past, present, and ongoing homeland of the Lenape people. The APS acknowledges with respect their continued presence and perseverance, and expresses its sincere thanks for the past and ongoing generosity of numerous indigenous communities and individuals who have offered their guidance, their expertise, and opportunities for collaboration. Oh, thank you. I don't know. Thank you. But yes, actually, for the Lenape, we can give a round of applause. Well, okay, that was good too. All right, so I'm going to start just very briefly with the founding. As Patrick mentioned yesterday, the APS was founded in 1743 by Benjamin Franklin and men like him who were looking for a place to exchange ideas. And our name is a wee bit confusing because while it was ideas, it wasn't ideas in the modern day sense of philosophical. It was the ideas in their sense of making society, communities, a better place. And I always love this quote from our founding document. Now that the first drudgery of settling the colonies, which confined the attention of people to mere necessities, is pretty well over, which it wasn't in 1743, but it's good to dream, we can turn our attention to loftier things. And the range of things that they turn their mind to is enumerated in this founding document. And it does include ideas about the natural world, what we would now call chemistry and biology, but it also includes things like viticulture, agriculture, transportation, roadways, all sorts of ways of applying science to make the community a better place, which is why I think Benjamin Franklin would be particularly excited about a conference on fieldwork. He was very much a hands-on scientist, and I can feel his spirit yesterday, and I'm sure that we will feel it again today. APS today. We have, as Patrick mentioned, 1,000 members, most of them located here within the United States. They gather twice a year in April and in November, so we are gearing up for the November meeting now, as well as hosting usually 2 to 3 conferences a year, and dozens of, like this one, and dozens of special events and lectures. I do want to give a shout out, as was done yesterday, to the APS Press in the back. Wave, APS Press. APS Press is looking always for publishing possibilities, so if this is something you're interested in learning more about, please do let us know. Our books are published in partnership with the University of Pennsylvania Press, and so are carried in their catalog website and other distribution mechanisms. And then, of course, the APS is known for its research. Over 300 grants are given annually, including to many of you who are here in this room. Because Patrick's not here and I get to spend a tiny bit more time on the library. So there are 14 million pages of manuscripts, over 325,000 books, the papers of 7 Nobel laureates, and an 8th one promised. And then an increasing number of digital and born digital objects, which is one of the future trends of archiving. And it's something that we are dealing with on a regular basis. Some portions of those archives are to my right and to my left, and so I encourage you during the breaks and during lunch to take a look. Among these are, on my right, your left, examples from our Center for Native American and Indigenous Research, including the work of linguist Mary Haas with the Tunica indigenous population. That research, so appropriate for October, by Ivan Sanderson, and to my right, your left, is Barak Blumberg, who of course helped to found the Lewis and Clark Fund and is best known for his work on hepatitis B, field notes from Luna Leopold on streams and rivers in Hawaii, and Barbara McClintock, Nobel Prize-winning geneticist. So I hope that you will have an opportunity to look at these documents at some point throughout the day. And if you yourself long to be in a vitrine, donate your papers to the American Philosophical Society, and you yourself might find your work, not you, these are small vitrines, you will not fit in these vitrines, but pieces of your work will fit in these vitrines, and you can be immortalized in plexiglass and in our research collections. Something to aspire to, something to aspire to. We do also have 4 centers, scholarly centers, that help to shape our work here at the APS. Those are our 3 major collecting areas: Native American and Indigenous research, early American history, and the history of science, which, since she's in the room, I will go ahead and say is the largest portion of our collection and the most heavily used. And it is, of course, the basis for the APS's founding. We also have the Center for Digital Scholarship, which supports all of our centers and creates an incredible array of online digital tools, including, for example, Mary Haas's work with the Tunica, which is now available in a fully online capacity, and a wonderful digital exhibit about Barack Blumberg. So the 3 more things, and then I will turn the microphone over. The first is, we very much want, it has nothing to do with the slide. The first is, we very much want the work in this conference, like all of our conferences, to live on. And so in addition to being the director of the Library and Museum, I'm also the general editor of one of our 2 peer-reviewed scholarly journals that we publish at the APS. The journal is called Transactions. So within a week or 2 of this conference, you will receive an email from me, Michelle McDonald, not spam, asking if you would be interested in contributing your work to a special issue, or 2, if there's enough demand, issue of Transactions. We publish quarterly. This issue would be targeted for probably mid to late 2027. So please do keep your eye on your email. We very much like to see iterations of this kind of work, which is wonderful in this 1.5 days, reach a broader audience whenever we can. We are also trying very much to get your thoughts and your ideas about your research in what we are calling lightning talks. How many of you have seen the lightning talk display? Look, your hands are like this because you don't want to admit it. How many of you have done a lightning talk? Raise your hands high. Look around at your peers. Exert peer pressure. Okay. We're doing this partly because this will be an activity that we're featuring next month at our semi-annual meeting, and so we're trying to get the hang of it. So we would very much appreciate your participation if you're willing to do so. Let me lay it out for you. There's a wheel, and you spin the wheel. It's kind of like APS's version of, what is that game? The Wheel of Fortune, except there's no cars and $1,000 prizes involved. Don't get your hopes up. Instead, you get to answer questions like, what does your research mean to you? And what do you think is the future of useful knowledge? And if you have no idea about either of those things, what is the best book you've read this year? Or you can make up a question of your own. We're really just practicing. So don't feel any pressure. This is all for fun, but your appreciation would be appreciated. And then finally, and related to this slide, the other activity that I would very much encourage you to take a look at is our museum. So our museum is located in Philosophical Hall. For those of you who joined us last night for dinner, it was immediately below your feet. It is free, it is open all day, so see the vitrines, see the museum, and learn more about what we are doing here at the APS. And then finally, Linda Musumeci asked if I would give you directions to the restrooms, because we didn't do that yesterday. Now, I apologize if you've been waiting since then. But given that you're fieldwork scientists, I'm assuming you're intrepid people and you may have already located them, but just in case, the men's restroom is located in the dungeon in the basement here of Franklin Hall, and the women's restroom is conveniently located just right outside this door. And the Wi-Fi is available on the back of the door and in your program. So with all of those logistical arrangements, welcome to day 2, and I'm going to turn the microphone over to Linda Musumeci to introduce some of our grant recipients.
Thank you, Michelle. And I feel so much better that everyone knows where the restroom is, because all of us forgot to tell you yesterday. I won't repeat what I said yesterday about the program, but as you know, we're celebrating the 20th anniversary. Well, you may not know, but this one gentleman may not know, but we're celebrating the 20th anniversary, and it's very exciting to move from the 4% funding ability we had initially to now we're up to about 23%. So it's quite a move forward, and it's been sort of slow and steady progress, and I just want to repeat or assert that we'll continue raising money for this program and adding to it as best we can, and I'll get to that in a second. And I want to reaffirm our commitment to the program, to all our programs, but to this one in particular. As academia changes, as the world changes, I think there's a particular need even going forward, and we're committed to it. So I want to thank everyone for coming. Also want to say that now that everyone is here stuck in a confined space, I can really go around and meet the people that I've been corresponding with for so many years, because you can't escape. So anyone I'd like to continue to, please come and talk to me. I really want to continue to meet all of you. She's not here, but I want to give credit to Joyce Marcus, who is a longtime chair of the Lewis and Clark Fund Committee. She brought us a lot of our reviewers, and she's very well connected to a lot of people in this room. So I just want to recognize the work she's done on this committee. I would invite everyone to provide, if you can, some feedback on the program, some suggestions for what you'd like to see going forward. I have ideas, but you're there, you know, you're in universities, you're in the field, so I'd like to have your feedback if you would share. There will be a survey after this conference. You could do it there. You could tell me if you have something on your mind. If you think of something on the plane, just everyone knows my email address. You can send me an email. Michelle brought out the invitation to meet with Allison in the back, and just wing it, you know, if you don't want to answer any of those particular questions. Really do, we encourage you to just share your thoughts. I love reading the reports that everyone sends in, but just hearing in your words and seeing, you know, the enthusiasm as you present the information, it's an altogether different experience. And that actually lets me transition to inviting 2 more of our grantees to come up and speak on what these grants have meant to them, and what their field research has meant. And so our first grantee is Cristina Torres, and she specializes in Spanish and linguistics at the University of Houston downtown, and she's in the Department of History, Humanities, and Languages, and she received a grant just this year. Here's Cristina.
Good morning, everyone. It's a pleasure for me to stand before you today. I want to express my gratitude to the American Philosophical Society for this opportunity. Your support has not only made my field study possible, but it has allowed me to grow both personally and professionally. Your commitment to advancing knowledge and supporting scholars is what keeps the spirit of discovery alive. Through this grant, I had the opportunity to conduct a sociolinguistic study in Cancun, Mexico, highlighting on the interaction between the Maya language and Spanish. My project explored how the 2 languages influence one another in daily communication, both oral and written. This experience allowed me to work directly with the speakers, document linguistic patterns, and better understand how language preserves identity and history. I visited the 3 states that make up the Yucatan Peninsula, that is Quintana Roo, Campeche, and Yucatan. I also had the opportunity to visit several archaeological sites along the Mayan Trail on the peninsula, taking pictures, videos, and conducting interviews and questionnaires. I came back to the United States so happy and grateful with the collected data that I will use I'm planning to write a book and to produce a documentary on the Mayan language and culture. Beyond the academic outcomes, journey deeply impact me in a personal level. Why? Because it made me feel more empathy, respect, gratitude, and the desire to support other people as I have been supported. Once again, I want to thank the American Philosophical Society and its donors for believing in us. Your support helps advance research to build bridges between languages, cultures, and community. I'm very grateful. Thank you so much.
And our next grantee is Devani Jolman, and she's a PhD candidate in ecological sciences at Old Dominion, and she's studying plant evolution, and she's a 2023 Lewis and Clark recipient.
Good morning, everyone. I just want to say it's been wonderful hearing from all of you and interacting with all of you in this multidisciplinary space. When I started as an undergrad, I entered as a pre-med student. But one of my professors recruited me to join his long-term project conducting floristic surveys of Southwest Michigan. And I remember standing one day in the mud of a bog that was newly discovered, and I had never felt so at peace with myself and connected to the system around me. And that experience with fieldwork was transformative for me. And now I find myself a fifth-year ecological sciences PhD candidate many years later. My research seeks to understand the ecological consequences of natural plant hybridization, and I do that by investigating wild hybridizing populations of blueberries across the eastern coast of Virginia. And if you've ever worked with hybrids, you know that finding and establishing a study system in the wild is very difficult and requires a lot of time in the field trying to locate them. And so I received the Lewis and Clark grant in my first few years, and it allowed me to spend 2 months searching, driving across Virginia, trying to find these plants with a few context clues. And it was integral to my entire dissertation. With the Lewis and Clark grant, I was able to find 9 sites, over 402 individuals, from which I collected 29 plant traits, leaf material for genetic work, and established a pollinator monitoring project. Every single chapter of my dissertation was relied on this grant. And I also, so it was foundational for my research, but through this grant, I was able to recruit over 10 undergraduate students to work with me. And so while the Lewis and Clark grant is foundational for great novel research, it is also foundational for the progression of science through the recruitment of students. Fieldwork was transformative for me, and I was able to extend that to many students who are now continuing on into plant science. And so I want to thank the American Philosophical Society and the Lewis and Clark grant for the opportunity not only for me to conduct research, but also to open the doors for many students to be able to experience the same level of excitement and connection with the natural world that I received when I was at that age. So thank you so much.
Thank you, Christina and Devani. And with that, we're going to start our first panel. And so I'm going to turn proceedings over to wonderful APS member and former Wenner-Gren head, Leslie Aiello.
Thank you very much, Linda. And I want to start off by extending more thanks to Linda and APS for organizing this meeting, and also wish happy birthday to the Lewis and Clark Foundation, because as we've heard, it's been so important to so many people's academic career and development. and long may it last. Okay, the topic for this morning is models for collection and preservation. And when I first saw the program, I was wondering, well, why, when we're emphasizing the practicalities of field work and all of this, are we going to talk about collections and preservation? And then the light bulb went on, because we're all collecting data. And as we talked about a little bit yesterday afternoon, it's our responsibility to safely and ethically collect these data, preserve the data, and also make those data available for future research and for public engagement. And it's actually up to us to do it, recognize what the problems are, and move forward with some solutions to some of these problems. Now, when I was thinking about this, I realized that I did my first field work 60 years ago. It's actually scary. I was an undergraduate student in 1963. And from that time, I've seen quite a development and evolution of the approach of the field to field work and data collection. And when I was a graduate student, nobody talked about it. Nobody talked about the ethics. Nobody talked about the difficulties of doing field work. We were all more or less dumped into the field. And I think the first realization that I had was when I was a PhD student in London. And I was working at the Natural History Museum there. And the human skeletons I was working with, some of them had cut marks on them, meaning that they were bodies that had been defleshed for collection. And this sort of began to light the bulb of ethics in my mind. But again, no one was talking about it. And it was before a lot of the interest and move for repatriation was considered. Now, as my career went on, there were a few other milestones that really, for my own development, were sort of the guidance as we move forward to where we are today with handling our data and preserving the data. And I think the first thing also is when I became head of the anthropology department in London in the 1990s. And it was at a time where some primatologists had been kidnapped in Uganda. And there was an issue, particularly in our own department, where one of our social anthropologists sent a female student into a war zone without any consideration of what the safety concerns were. At that point, there was no review of projects. It was a personal development of research between an individual and their student. And as head of a department, this really scared me. And I set up a system of review for our PhD students that would involve a broader group than just the single PhD supervisor, and also instituted risk assessment. There was a lot of pushback against this because there was a feeling I was interfering between the special relationship between the advisor and the student and all. But it was a safety issue. And as my career moved on, I became head of the graduate school in London. And at that point, there was no IRB system for any social science research. And this, all the red lights and bells, went on. The only way that you could get IRB in the UK at that time was through the National Health Service, and it was all clinical research. Now, we set up a IRB for social sciences. There was tremendous pushback from the 72 departments at the university. Again, we were meddling in research. But what saved the day on that was some very high-profile problems with social science research at the time. And the Research Councils, the equivalent of the National Science Foundation and the National Institutes of Health, slapped a ethics question on their grant application. And I suddenly became everyone's best friend. And what I'm happy to say is that system, it became a model for IRB boards for the social science in the UK, and it's going strong now. But the reason I'm mentioning these things is all of this has happened in a relatively short amount of time. In my career, as seeing things develop. And so some of us as researchers think it's meddling and it's too much work and all. But if you think of the advantage of this, not only to ourselves as researchers, but also to our research participants and the field in general, it's tremendously important to give thought to these things. And when I took over Wenner-Gren, and for those of you that don't know, Wenner-Gren is one of the main funders for anthropological research. And I would give out about $6 to $7 million a year to the field. So I saw a lot of applications. And what was interesting during my over decade in running Wenner-Gren was seeing the transition to engaged research and the necessity of providing funds for involving collaborative research, involving the research participants, and also engaging with them for the to bring back the results of the research. And we had a engaged anthropology grant that actually gave funds for our grantees to go back into the field to share the results of their research. And this now is morphed into a big program that supports collaborative research that is developed and carried out in conjunction with the local communities. Now, my last step in all of this is a current involvement I have with the National Museum of Natural History. And it's really been a golden opportunity to see the problems of running museums, maintaining collections, making them available, and just keeping everything afloat. Now, what we're going to do is cover all these areas in the next 3 talks. And we're going to start with museums. And our first speaker is David Skelly, who's the Frank Osler Professor of Ecology at Yale, and also director of the Peabody Museum. And in real life, he researches amphibians and the relationship to climate change. But David's going to talk about the future of natural history collections. And he's done the paper in conjunction with Kurt Johnson, who's the Sant director of the National Museum of Natural History, the Smithsonian Museum. And David's going to be followed by Alex Baker, who's an anthropological archaeologist and past president of the American Anthropological Association and director of the Arkansas Archaeological Survey. And he's going to talk about useful knowledge and its perils, open data, sovereignty, and the problem of cui bono. Now, he's going to focus a lot on the relationship with Native Americans and I think it's basically who has control and who has the sovereignty over the data. And our last presenter is going to be Ted Schurr, who's professor in the Department of Anthropology and head of the Laboratory of Molecular Anthropology at Penn. And he's going to talk from a personal point of view on the challenges of managing samples, data obtained through field work, perspectives from biological anthropology, and particularly from anthropological genetics. So we'll turn it over to David.
Thank you, Leslie, for that great introduction and setup. And thank you to Linda and everyone at APS for setting up what has been a wonderful meeting, and looking forward to what goes on today. One of the parts of Leslie's setup was mentioning my co-author for this talk. So that's Kirk Johnson, getting some accolades there for an award he got. I think he deserves some accolades right now for overseeing a Smithsonian museum where the last 9 months have been pretty special. And now, having to deal with a shutdown, although Leslie, who sits on the board, says that he's enjoying the quiet, evidently, at a museum that... It's all relative. It is relative. I mean, they get 6 million visitors a year, so it doesn't take much to be quieter than that. But this talk that I'll give is the distillation of many conversations that Kirk and I have had over the last decade or so while I've been the director of Yale's Peabody Museum. Kirk actually sits in this circle of board membership. He's on the Peabody's board. He's a Yale alum. What I'm going to do is talk about the future of natural history collections at a moment where, as another person I have to say thanks to is Scott for setting up some of the challenges that museums and collections have been facing. And does this work? So should I just say next slide? Great. So I'm going to do this in 3 parts. I'm going to talk very quickly, because I'm ticking down here, why do we have natural history collections? And I'm going to give you the view from where I sit. What do collections in decline, what does that mean? And the sharp-eyed among you will recognize that one of the slides that I'm going to show is exactly the same as one of the ones Scott showed yesterday. And I'm going to talk about one take on some ways that collections can have a vibrant future. And on the screen here is one of my doctoral students, Julia Portman. The word that I'm using today is collections, but collections happen because of collecting, and collecting is fieldwork. And to have vibrant museums, to have vibrant collections, you just heard here at APS a pitch for building collections. Collections that don't build die. Their vibrancy, their integrity comes from active curation, active use. They were created by people who wanted something out of them. They will be maintained by people who are getting something out of them. Okay, so I did all of this thinking about the talk that I'm going to give today in the aftermath of having spent about 10 years thinking about the future of one particular museum, Yale's Peabody Museum, and planning and then carrying out a renovation and expansion of a natural history museum in an era when that is not necessarily the norm. So what you see on the left is from a BIM. Some of you will know a BIM is a building information model. That's how architects work these days with big complicated projects. And that's a rendering. You could put on one of those headsets and march around inside this virtual building. And then the picture on the right is actually from a couple of years ago when we were in the middle of building this museum. And in between there and before even that image was created, we did a lot of thinking about what should a museum look like in the future. What does that mean? What are we trying to accomplish? So back to the question of why do we have natural history museums, I'm going to give you the answer from my institution at Yale University, and we can go back to a very specific moment in our institution's history. So the fellow in the painting on the left is James Dwight Dana. Among other things, he was called America's Darwin. He was the naturalist on the U.S. exploring expedition that was inspired by the Beagle voyage. And he was also the second scientist on the faculty of Yale University. The first was Benjamin Silliman, and Dana was his protege. He has something that, as a scientist, I always say all of us, or at least I would love, and that is that one of his publications, whatever, 130 years after he died, is still in print. His system of mineral taxonomy is used all over the world. He was a brilliant scholar, worked on all kinds of things, and in the 1850s, when natural philosophers were starting to call themselves scientists, he gave a talk at graduation in 1856 on the future of Yale, what was then Yale College, describing a future where Yale would become a university. And to do that, Yale was going to have to embrace the sciences. And to do that, it was going to have to create a museum. That was integral. A museum is a Victorian startup. It was a way to embed science and technology into what was a pretty fusty institution. I have Yale's catalog is, even though it's digital now, is called the Blue Book. And I have a Blue Book from even before they were blue, from 1844. And if you take a look at that, what was said about Yale of that time in the middle 1800s is that you could have dropped a Roman centurion into the classroom, and not only would they have understood the material, they would have understood the language it was being spoken in. And it wasn't for, except for efforts from Silliman and Dana, that the modernization of the curriculum started. And they started almost immediately after this talk awarding North America's first PhD degrees, establishing a graduate school, and eventually becoming a university, and very quickly after this, establishing a museum. So I've heard this word discovery a couple times, even this morning and yesterday, and I use it advisedly. I know this is a word that carries a lot of weight and freight, but what collections are substrate for learning, and this other word, discovery. In other words, the way that I'm using it here is we learn things about the world that we didn't know because collections exist. We've heard people talk about this already. Scott did a great job last night of giving some examples. And so on the upper left here, we have some osprey eggs. These eggs from the species that were collected over a long period of time. These, I think, are from 1913. Were used to document the changes in eggshell thickness that led us to conclude that DDT was having these terrible effects on birds. The quagga up on the right is an example of a species that was used to develop the technologies around ancient DNA. And those specimens, of course, came out of museums. And the middle and the bottom here is Yasmina Viman. Yasmina got her PhD at Yale in 2021, working with the collections. She was responsible for research that helped us understand that dinosaurs had colored eggs. In general, she is developing, and I think she would hate me using this word, but sort of paleobiochemistry. She's figuring out how ancient organisms worked and developing the technology and the means to interpret the results from different technologies that allow us to say, for instance, in some of her more recent work, that many lineages of dinosaurs were warm-blooded. And that was one of the biggest open questions in paleontology. So brilliant stuff that all was based on collections and museums like the Peabodys. This is the figure that you've seen before, so you should all have this memorized by now. So great stuff, right? So what's up? Why am I saying museums and collections are in decline? And it's because they are. Kirk, more than probably anybody in the country, and I think we have a curator here from the American Museum, they get phone calls, emails, texts all the time saying, we're going to get rid of our collection, we need your help. The number of museums is going down. In general, the number of curators is going down. The amount of collecting activity is going down. Some of that is because the fields that developed these Victorian startups, like taxonomy, are not what they once were. There are fewer people pursuing that, there's less training, and there's a lot of hand-wringing about that. But what hasn't happened, and what I'll say more about, is we haven't picked up new fields that may be quite interested in using collections at a rate that I think we need to. The other thing that's gone on that we've heard about is, collecting has a really fraught history. And part of the legacy of that is that people aren't as interested in necessarily having people come and collect. One of the people that showed up on the list of researchers for the Fieldwork Symposium that's got listed is Michael Donahue. I had dinner with Michael last week, and after working in the highlands around Oaxaca for 50 years, he got cut off. Not because of anything he did, but because some ag researchers went in there and grabbed some tissue from some corn plants, and they're trying to develop technology to improve corn cultivation, and they didn't do what they were supposed to, and so that kind of activity has a blanket effect. And this article and many others are being written not by Americans doing what we heard called helicopter science, but by people who in country are not able to do biodiversity-related research in their own countries. And that's because when the Nagoya Protocol was set up, each country could develop their own procedures for permitting, and it hasn't gone beautifully. And there's less biodiversity research going on. There's also not a lot of access and benefit sharing going on. So we're stuck in a weird place, and it varies greatly by countries. I'm not saying there aren't good examples, but this is something that needs attention. In the middle of all this, we had Duke do what I call a sidewalk announcement, and that's when somebody says, we're getting rid of our collection. They haven't talked to anybody, and then our phones start ringing off the hook. Dave, what are you going to do about what Duke just announced? It's not my fault. What are the people at Duke thinking? The year before they announced they were getting rid of their herbarium, they declared themselves the climate university. They're sitting on one of the greatest resources to understand climate change, and they're putting it on the sidewalk. It's been a mess. Sorry if there's any Duke alums. So what does the future look like? The future needs to be different, and that thing is flashing. I got to stop. But we have ways of moving forward that are going to involve reaching out to folks who maybe don't see themselves traditionally in a museum sense. There's a couple of examples here. One of them is from the Peabody. We have a 15 or 20-year effort to document Shifaka in southwest Madagascar that Allison Richards started. That's a virtual collection. It's all digital. Lower right, the Australian Museum came up with an app called Frog ID. And they've had over 1 million submissions in a country that has 1/10 the population of the United States. It's changing what we know about frogs. I'm a frog guy. And it's also changing how much people care about frogs because they're hearing about it more. So we need more of that. And I'll leave it there.
Okay, our next speaker is Alex Baker on useful knowledge.
Good morning. I'd like to begin with a general premise. Fieldwork always generates collections. Sometimes those collections are objects, artifacts, or specimens. Sometimes they're vouchers. Sometimes they're data or measurements. But all are collections whose use life continues long after fieldwork ends. Who owns those collections, and more importantly, the data those collections represent or that may be abstracted from them, is an increasingly complicated issue. Permission to collect or title to objects doesn't necessarily convey rights to future use or to the economic benefits gained from their study. Who benefits, cui bono, is a growing issue where useful knowledge is concerned, especially in the context of indigenous or descendant community rights. See if I can make this work. There we go. In an ideal world, all would benefit, and the rising tide of knowledge would float all boats. The FAIR principles arising from a 2014 conference in Leiden are constructed on this logic. They call for all data to be findable, accessible, interoperable, and reusable. Taken at face value, such principles seem inherently well-fair. The results of scholarly inquiry should be findable and accessible, and in a digital world increasingly dependent on linked open data, accessibility absent interoperability is neither efficient nor adequate. And the reason for all three of the prior principles is to make data reusable, or phrased differently, to perpetuate its usefulness. The simplicity of these kinds of deontological principles is alluring. But in some instances, findable and accessible data are problematic. The CARE principles were developed to address the tension between benefits of such fair sharing of knowledge, on the one hand, and on the other hand, concerns within indigenous communities for both protecting indigenous rights and traditional knowledge and limited opportunities for benefit sharing. The CARE principles call for collective benefit, authority to control, responsibility, and ethics, and are designed to temper the FAIR principles with respect for indigenous rights and self-determination. Several initiatives seek to combine FAIR and CARE concerns to ensure that sovereignty is respected and indigenous communities benefit from work involving them or informed by traditional knowledge and practice. But that's easier said than done. One challenge is overcoming what we might call the surjective fallacy, namely that there's a one-to-one relationship between data sets and communities with rights to or over those data. Sometimes a single group does have dispositive rights over a data set, but the closer one looks, the rarer that proves. That surjective fallacy has a subjective correlate. Because that assumption is fallacious, researchers understandably end up making subjective decisions about how best to proceed. In the best case, they make a good faith decision regarding whose sovereignty should be given priority and most weight in this particular instance. In the worst case, they use that difficulty in assessing the relative merits of such claims as an excuse for respecting none. This was the concern Native groups expressed about the original NAGPRA regulations. Second, and perhaps third, are presentist fallacies of at least two kinds. One is that the present is an adequate representation of the past, ignoring histories of dispossession, displacement, and past population dynamics. The trickier presentist fallacy is that the present is an adequate representation of the future. Collections and even more of their associated data perdure, and their significance changes and ideally grows over time. Permissions given or rights granted are subject to change, and because intellectual property rights are tied to data about collections rather than the objects alone, having the right to collect samples doesn't confer rights to their use or benefit, or the future benefit extracted from samples or collections. Such rules are well established, albeit not always followed, in biomedical research, which is less subject to the surjective fallacy, but they're less frequently discussed for the field sciences, excuse me, where reuse of collections is expected and generally desirable. Consider an example. This bag was recovered in 1932 from an Arkansas rock shelter and is roughly 2,000 years old. It contains quinopod seeds representing fully domesticated cultivars combining naturally selected adaptations to the American mid-continent and human-selected traits increasing economic value, such as an increased volume of the endosperm. And as a result, such seeds have enormous commercial and agroeconomic potential. But who owns the rights? And who should benefit in the event of commercialization? Based on geography, we'd likely affiliate the bag with the Osage Nation. And I have no doubt that communities ancestral to the Osage participated in the domestication of Eastern agricultural complex crops. But Osage tradition and archaeological evidence alike place Osage arrival 1,000 or more years after this bag was deposited. Matters become still more complex if we consider the extent of sites with similar evidence for the same domesticates. Domestication took place over thousands of years and across much of the American mid-continent and was not necessarily associated with any one group. So who should benefit? None of the three commonly employed responses seem to meaningfully accord with care and fair principles while also offering meaningful respect for indigenous data sovereignty. Given the complexity of identifying a single group, one might decide all such groups should have equal rights of consent. Essentially, any one group could then veto the decision of the remainder. Alternatively, on the basis of high aseity, one could affiliate each individual example with a given community and assert that they should solely benefit and should have dispositive rights. Beyond the practical effect of allowing one group to solely benefit from collective knowledge, this logic would allow a commercial applicant to set up a variant of the prisoner's dilemma. where whichever group offered the most advantageous deal could disenfranchise the remainder unless all refused to break ranks. Or one might throw up one's hands, deciding absent a simple method for recognizing rights, none should be assigned. I phrased this problem in terms of archaeology, but these are questions of growing concern across all field sciences. Last year, the World Intellectual Property Organization adopted a treaty on intellectual property, genetic resources, and associated traditional knowledge, which affirms that anyone seeking to patent genetic information reflecting traditional indigenous knowledge must acknowledge its source. On its face, a win for indigenous communities. But if the applicant doesn't know or can't establish the source, they can simply make a declaration to that effect. It isn't meaningful to assert that an indigenous community should hold all rights to all data, not only on grounds of practicality, but because that necessarily impinges on the rights of other indigenous communities. Multiple groups can simultaneously hold dispositive rights over the same data. But attempts to allocate those rights in advance require categories and definitions that privilege one set of constructs over another and assign greater functional sovereignty to some groups at the expense of others. It's not simply a matter of multiple claimants, but of disparate frameworks for assessing those claims, and of multiple understandings or constructs regarding what kinds of data are subject to claim or control. We also need to think of rights and collections in more dynamic terms, as having not just a past, but a future. Issues of rights and permissions are often and necessarily negotiated at the outset of a project, but once recovered, collections and data persist and are ideally reused. This is where principles derived from biomedical ethics serve us poorly, both because of the surjective fallacy, which again is less of a fallacy in biomedical contexts, and the presumption that the reuse of samples and data is the exception rather than the rule. How does community-engaged fieldwork maintain engagement once fieldwork ends? Here I'd like to explore one mechanism for addressing those challenges without inadvertently, or perhaps intentionally, placing the researcher in the position of arbiter. Local contexts, notices, and labels, a set of conventions developed for indigenous data sovereignty. Notices are standardized icons stating an individual or institution's willingness to engage with indigenous communities. Their core semantic content is set and standardized, and they can be applied by any institution or researcher subscribing to local contexts user agreement, here by my home institution. These are the notices. And that's the text for one of them. Labels, by contrast, are developed by and for indigenous communities. How much or how little to disclose is up to the community, and they can also use labels to identify handling or other protocols to be observed. Traditional knowledge labels relate to attribution and provenance, cultural protocols, and permissions. Biocultural labels extend the same assertion of rights and intellectual property claims to biological and genomic data. Labels may be customized to meet the needs and constructs of a given community instead of assuming that one framework works for all indigenous groups. They have three other important advantages. One is that they offer a more nuanced and granular set of protocols for access. They also allow indigenous groups to clearly articulate their claims to data or intellectual property in a highly visible and recognizable manner. In circumstances like those envisioned in the WIPO Treaty, applicants would know immediately which communities asserted what kinds of rights. Of equal importance, the notice and label arrangement resolves or at least obviates the surjective fallacy. If more than one group can make claims or has associated knowledge, more than one label may be associated, and those labels can be updated over time. Labels are hardly a panacea, and three shortcomings must be acknowledged. First, they're not intended to apply or to be used for non-indigenous groups. From an institutional standpoint, and to simplify fair application, it might be preferable to have common indicators denoting traditional knowledge and data rights associated with descendant communities of all kinds, instead of limiting the scope of the notice to indigenous claims exclusively. Second, in a time when many indigenous communities are overstretched and facing myriad demands on their time and resources, development and application of label content may be potentially beneficial, but practically burdensome, particularly in the short term. And finally, labels don't intrinsically enforce or enact whatever controls or restrictions an Indigenous community may apply, although they may have significant evidentiary value should enforcement be needed. Use of notices and labels does not directly solve the challenges of competing claims or contested control, but adopting conventions that allow Indigenous groups to tag data with their claims and access protocols, especially in a manner that acknowledges multiple claims or constructs without choosing among them, offers greater potential for equity. It also prevents field scientists from playing the role of arbiter of competing claims and choosing among sovereignties, or worse, playing the learned fool, and thereby allowing useful knowledge to become abuseful. Thank you.
Okay, thank you very much, Alex. And our last speaker, Ted Schurr.
I'm delighted to be here to be able to contribute to this conversation on field work. It's been very stimulating for me to think about it from a multidisciplinary point of view. I'm going to offer some perspectives on this from the standpoint of biological anthropology, emphasizing genetics, anthropological genetics, but trying to draw other areas of biological anthropology into the conversation as well. So my talk is going to follow this outline you see here. I'm going to give you some images from field work we've conducted in the past and the work that's ongoing. That provides the context for doing the sampling, for doing genetic research or biological anthropology research. And so I'll talk about the types of samples and the data we get from them and the various constraints and issues that arise from that. I'll then mention at least something in passing about the new field-friendly methods, which are allowing us to do more of the work in the lab in the field. And then turn to one example of work we're doing with community-engaged work with communities in Charleston for the Anson Street African Burial Ground Project, and then I'll offer some concluding thoughts. So with regard to field work, I first want to acknowledge all the communities that have been involved in our research over a number of decades. Without their involvement in this work, their support, their interest, none of the work that we've done and published would be possible. And what I'd like to do in the following slides is in various ways talk about places we've worked, people we've worked with, and some of the issues that have arisen in the process of doing the work, which have informed how we talk about the genetic information that we gather, the context, the interpretive framework that we use. So the first photograph here are a mother and daughter. Ka'u is the mother in the red dress, and the daughter, Nina Nilva, was a nurse who worked with us when we were doing research in Kamchatka on the history of Koryak and Itilman populations. And very supportive community, and I also, in this case, I want to also mention the fact that that Ka'u is actually a Nimilan Koryak speaker, which is a growing indigenous language. And part of the reason why it's diminishing is partly because of Russification and the way in which the communities have been treated since the 17th century colonization, and these legacy effects you see in all sorts of ways in modern day communities. We've also worked with elders in the Seekonk Wampanoag tribe in Massachusetts. This is Three Bears. Very friendly man who gave us lots of insights into the tribal history, and they were interested in actually in exploring this using genetics as a means of reconstructing their tribal history. We also have done a fair amount of work in other parts of Siberia, including the Altai-Sion region. My colleague is Ludmila Osipa is on the far left. We work with Kumadan groups who are part of a number of Turkic groups that are connected deeper time to ancestors of Native Americans, which is why we and they were interested in working together. Sadly, both the matriarch of this community and also in red dress and Ludmila Osipa have passed away, as have other colleagues in Russia that we've worked with over the years, and also the other elders you've seen in the past. So this is just a reminder of the loss of knowledge that comes with people with this knowledge dying, and including people doing field work for a number of decades. Part of the way we're trying to deal with this is to collect field information on the histories of communities where we work, as we are here in Mongolia, Georgia, with my colleague David Chadanava. At the same time, we're also gathering genealogical information from the communities we work with, individuals, both to help interpret the genetic information, but also to learn about their own histories. In the case here with Sharina Lasvili is talking to 2 Armenian women who live in the community and whose families actually moved into Georgia after the Armenian genocide in Turkey. We've worked in a variety of places in the Caribbean as well, in this case in St. Vincent Island, and here with Garifuna communities who are very deeply interested in their indigenous past. This work is done collaboratively with Jada Bentor, as you see pictured on the left. A major issue for these communities is actually getting recognition as being indigenous communities or descendant communities and seeking reparations from the British government for their dissolution of the Garifuna communities and dispersal of many across the Caribbean area once they defeated Garifuna forces in the 18th century. We also have done a lot of work in Turkey and then this area of West Asia. Here, Timur Gulikin is taking consent from these Turkish men at the tea house. And I wanted to point out here that one of the things that was about this project, which had some constraints or had us to refigure our projects, was that all samples had to remain in the country, all work had to be done in the country, nothing could be removed because of the permissions restrictions that were in place. Also, I want to mention that all these projects took 2 or 3 years in terms of consultation to actually get going and be able to undertake. We're also fortunate in doing this field work to be able to see various expressions of cultural celebration and a reclaiming of identities for various communities. Here we have a Nuvialu dancers at the Circumpolar Games of 2010 up in Nunavik in Northwest Territories. Fantastic dancing. We've also were working with, through the Baikal Archaeological Project, working in Hokkaido, Japan, doing comparative work for Neolithic communities. Here at this site in Funadamari, being blessed by priests from the Ainu communities who are reclaiming this territory as their traditional lands and trying to exert this vis-a-vis the Japanese government to have more control over this territory. And then finally, for an example from fieldwork, we work with Bermudians in the St. David's Islands. The islands weren't settled until the early 17th century, but these communities trace their roots in part to the remnants of Wampanoag tribes who were defeated in the St. David's War, or King Philip's War, thank you, and then sent to Bermuda. And so on a biennial basis, they have a celebration with tribes from the Northeast to come down and celebrate their heritage together. So again, all this forms the background for the genetic research that we want to do. And we use a variety of samples for this, especially for genetics, including blood samples, buccal swabs, spit samples, and then of course other work that may involve biological anthropology includes breast milk, urine, feces, hair, and also blood spots. And so each of these types of samples require certain kind of handling and considerations. I'm sure many of you know this already. But, what is the extent of invasiveness? And in this case, there is a decreasing use of venipuncture for field-based studies. What kind of materials and methods do you need to use? Well, each of these samples requires different kinds of acquisition materials. Different cryogenic needs. How do you have to keep them cold? And how do you do this, taking things into the field? How long will things preserve at room temperature in other places, especially in more remote areas where you may not be able to get a helicopter for 2 weeks? Their downstream analyses are multitudinous. You can sequence, you can do immunoassays, microbiome work from the feces samples, and then different kinds of data that are generated in the process that then require you to handle them in different ways and analyze the data in different places. And especially if you want to do multi-method kinds of analysis, then integrating all this work can be pretty complicated. On top of this, it's now possible to get data from ancient DNA sources of various kinds, especially tooth and bones from humans, but also from coprolites and residues of various kinds from some other materials that give you insights into the history of the diet of various persons. And a constraint here, of course, is you have to work in an ancient DNA lab to carry all this work out. All the process of doing the prep work to be able to do any kind of sequencing has to take place in specialized places, which aren't that numerous in some respects. Also, some of our work indicates, there's a lot of emphasis on using similar clean suit type of approaches in the field to remove materials for analysis. In a way you see here, my colleague Raquel Flescus is working on some of our research samples from the Chesapeake. What makes all this work possible is the fact that new technology allow you to generate huge amounts of data, which has already been referred to here in talks before. This is a challenge. I mean, it's great that you can have it, but then there's a matter of analyzing all of this information. But you get to the point of data management, and where you are doing all the analysis of the samples you've taken in the field, you have all the bioinformatics work to make sense of all this, and then there are various ways you can actually begin interpreting this in different kinds of ways for different questions you're answering or trying to address. This is references biomedical research, but it applies equally to biological anthropology research. Moreover, from the same communities or based on different projects you might do, you can potentially analyze aspects of genomic variation. You can look at transcriptomics, which tell you something about how genes are expressed. You can look at epigenomics, which actually may reflect lived experiences and social cultural impacts on the biology of people you're working with. And if you're collecting fecal samples for microbiome work, you're trying to get insights into aspects of health and disease vis-a-vis the bacteria in our guts. Or in the case, actually, of the next slide, simple dental calculus, I'm going the wrong direction. Simple dental calculus from teeth allow you to get all sorts of details about diet and health and oral periodontal characteristics of populations that come from food particles that are trapped, the bacteria, proteins that are there, all new ways of thinking about what people did in their lives. One thing that may help some of this going forward is the fact there are newer kinds of field-friendly tools that allow us to do work in real time in the field, like these handheld sequencers. We generate thousands, millions of base pairs in a matter of a day or two, and actually be able to share that with the communities you're working with. The final thing I'd like to talk about is actually our work in Charleston with the Ancestry African Burial Ground Project. It was initiated by Aiello, who you see on the left, and when the discovery of 18th century enslaved African remains were disinterred when the cultural center was expanding a wing in the middle of downtown Charleston. Through engagement with the community here, we built a project to be able to begin to analyze the remains of these individuals with community guidance and approval, so that we can then carry out a project, this multifaceted project that you see outlined here. Central to all this work were the questions they were asking us and asking us to try to answer through this kind of scientific research. We shared all this research with them at each step of the way and continued to come back and get feedback and ask for permission to go forward with work beyond the initial analyses. This also led to a significant reburial of these individuals at the site where they were found, including, this is Aiello here, and then the mayor of Charleston, who was also important in helping make this work go forward. And in parallel to this, we actually did DNA testing of the descendant community members who wanted to actually try to trace their connections to the ancestors and learn more about their own ancestry and connections back to Africa, many of whom have never had an opportunity to do this. So we carried out this work in a couple of phases. It's still ongoing, and each time that we get results, we report back to them all the results we have and talk to them, and all the conversations we have about the project, both from the ancient DNA side and the modern DNA side, will inform the publications that we create and we publish. So let me now conclude with a few thoughts based on all these ideas here, and in part inspired. I changed the slides a little bit after hearing everyone yesterday just to add a couple little points. But in any case, one of the things I wanted to mention here that's really crucial for preserving all the data we get from the field, photographs, videos, and so forth, even in current projects, and they accumulate extremely rapidly. And so, of course, places like this are areas where you can archive it. But we find ourselves challenged to actually handle all the materials we're getting from our field work across the world. We also now have new ways of expanded possibilities for sampling, especially with non-invasive methods. And the open ancient DNA opens up a far number of vistas to actually understanding human history in a variety of different kinds of ways. These new analytical methods, of course, generate huge amounts of data, which then become challenging to actually assimilate and aggregate and make sense of what we have using newer computational tools. This integration is very important because it's this complex picture that emerges when you use this multidisciplinary approach, which really supersedes anything you can do with one method versus the other. And so we try to do this kind of work going forward, although of course it's time consuming and requires a lot of coordination with various stakeholders who are part of the projects. And finally, I want to mention, though, that it's been alluded to in various places, the ways that the work we're doing now requires new kinds of thinking about consent and approval. Often it's long-term and requires a lot of investment and time in building relationships with communities. But, you know, community engagement is central to these projects. And I also want to emphasize that consent in this context is ongoing. It's not a singular moment. And furthermore, what happens, and there's not time to talk about this, but it was, you know, what do we do with reconsent for samples going forward? This is yet another stage of work, especially with human samples, which is really crucial, but it requires yet another step of the consent process of time and energy to make that happen, although in the end, the work that is created from this is really quite extraordinary. So let me conclude simply by thanking you for your attention with a picture here of the Caucasian Plains in Georgia, and I'd be happy to answer any questions you might have. Thank you.
Okay, we have plenty of time for questions. And what I'd like to ask you to do is when you ask a question, if you could identify yourself and where you're from. So, Scott.
Thanks very much, Scott Edwards from Harvard University. I'm curious, just from the last talk with sort of modern anthropological, biological samples, what's the typical practice today in terms of archiving them, or are they sort of destroyed once the project is done?
The microphone is on? Yes. So the process we follow in the consent process, informed consent we do with individuals is outline the use of the samples, the duration of the sample use, and what will happen to them after the fact. Ordinarily, the endpoint for all of these samples is to be destroyed, and then identifying information removed from our data sources and things of that sort. We also give ourselves the ability to go back to ask for extended consent to do other kinds of analyses which emerge out of the work we did initially, and then inform them about this and to make sure that there's approval to go forward, so there's a longer legacy of that work, of the initial analysis. And of course, they're stored in refrigerators and freezers and in secure places so that everyone knows where the work is being done. They can actually come to the lab and see the samples and things of this kind. So ultimately, though, the disposition of most samples, these biological samples, they'll be destroyed and no longer available for use or sharing of any kind, which actually we also explicitly indicate that's not part of what we do. Everything we do is controlled by us, done in-house, or work in coordination with participating testing labs, which give us the information that we share with them. That's part of our arrangement to share our knowledge about the work.
Thanks to the panel for a great series of talks. My question's for you, David, and I'm Jordan Karoubian from Tulane University. As a curator of a museum, I'm just curious about how you view the transition and the increasing inclusion of digital material as opposed to real specimens. That's something that you highlighted as a future direction, and it certainly seems to be going that way, but there's obviously costs and benefits and complexities involved with that, and I'm just curious if you would talk a little bit more about your thoughts on that.
Am I live? Yeah. So great question. Part of the answer is that we stole someone from Tulane. Nelson Rios is our head of informatics in the museum, and he's really helped us move much faster than we used to, having a unit that he oversees that is pushing forward on all kinds of initiatives related to access, creating new forms of digital data, purely digital collections. So, when as an ecologist, that was not a big leap for me. I'm not coming, I've been told, well, you're not really a museum person so many times by people who come from these more traditionally associated fields with natural history museums. And I guess one of the ways that I'm not is I never had any discomfort with that at all. We, even before Nelson joined us, we had a tradition like that Lemur database of embracing digital forms of data. That was really, that project with the Shifaka was where we kind of cut our teeth and figured out, what does that even mean for us to have a purely digital collection? How do we present that? we're doing lots of work with some of our anthro colleagues who work in contexts where they cannot bring material back to the United States. And so they're curators in a museum. They are never going to contribute to the material collections of the museum. And that's going to happen more and more. We have to be comfortable with that. So Jessica Thompson is an example. She works in Malawi. Malawi does not have a national museum. There's kind of a thatched roof hut where all the samples in Malawi live, and her husband happens to be a pretty handy guy, and he's built the cabinetry where her specimens are kept there. And we're working with folks in Malawi to provide the digital support for collections that will never come to the United States. That's the new normal. I mean, that's what I mean. We have to be leaning into being useful to people who are interested in collections in whatever way we can, and not be totally hidebound to older models. That is a way to end up on the sidewalk, right?
As a follow-up, what about the consideration and the advancement of digital twins for artifacts, specimens, and other things that have a lot of complexity? And the data principles have a lot to do with this as far as interoperability, reusability, exchangeability.
So digital representations of objects that are available online. Yeah, and in some cases, those digital twins are better than twins because they can show, depending on the technology that's used to image and otherwise analyze them, they can show aspects of them that aren't visible otherwise. So we have a couple of curators who are adding more terabytes than I care to think about using a micro CT scanner. So, you know, fossil preparation today looks a lot different than it did even a few years ago. And in a lot of cases, we're leaving especially fragile material or material that's in context where there may be, you know, the matrix may hold the story. We're leaving fossils jacketed, scanning them, and the digital twin is the representation of the fossil unless there's some extraordinary other use request. So that's also part of the future, I think.
And if I can follow up with that, the bag I showed you is 2,000 years old. We don't normally handle it, but micro CT scans have allowed us to see the internal structure of the bag, understand more about what's in it, and we no longer have the bag circulating and being used. If someone's got a question, we can normally get the data from the twin or from other kinds of analysis that have been done. Many bags like that were found. Most of them were cut apart so people could see what was inside, which isn't ideal.
I have a follow-on question for Alex. What's the opinion of the indigenous people in relation to digital twins? Because I've heard in some instances that they have the same ceremonial import as the real object might.
And Leslie, I think you already know what my answer is going to be. It depends on the group. There's a wide variation. As part of implementing NAGPRA, one of the things that many of the groups we work with have requested is that we provide digital twins so that they, without having to travel, especially so elders don't have to travel, can assess collections and get a better sense of things which the institution has not already identified as being NAGPRA related. Because unfortunately, the way the regulations were set up, the museum makes a first determination of what is covered under NAGPRA. And museums are not necessarily the best judge of that. But by circulating digital information about collections to groups very broadly, they can make that call. They also will then make a call over how broadly that digital information will be shared afterwards.
I don't know. Perfect. Thanks so much. Super interesting talks.
Yes, and please identify yourself.
Yes, my name is Devaney Jolman from Old Dominion University in Virginia. Working with herbarium specimens was integral in my undergraduate career, and it still is as a PhD. But I find that my training in taxonomy that I received in my undergrad is abnormal for many other graduate students. And there seems to be a downplay in that hard skill of using a dichotomous key and going out and spending that time doing taxonomy because of advances in technology using apps to quick ID in the field. How do we move forward? And I want to say using the apps makes field identification more accessible for the public. I 100% agree with that. However, how do we move forward with this wanting to embrace the use of this technology, but also encouraging this hard skill of taxonomy, and I believe that that is in relation to the kind of reduction in field collections. How do we integrate both of those things as we move forward?
So great question. So, you know, I'm going to build on a conversation I had last night. I think the best way to view this is if we position ourselves in museums and collections and doing field work, like, no, you got to do it the old school, uphill in the snow both ways. That's not going to broaden the tent. These tools are actually amazing. They're not perfect. And anybody who works on a group can tell you of instances where you get weird things out of an app that are not right. However, especially for people who are just starting out, it's a tremendous boost. You know, you don't have to have, not everybody out there can have an expert on their shoulder, right? And this can be sort of that expert on your shoulder, and it can get people started to get to a level that maybe you're at in your PhD work. You have to get beyond what an app is going to be able to help you with. But I think that we should be leaning into it, and that's why I use the example of the Australian Museum's promotion of Frog ID. I did my postdoc in Australia in the 90s, and you know, frogs were not on the top of list of things that people thought they should be spending time on. And when some Americans showed up saying, I work on frogs, you know, I still get that, but you know, people are like, really? You came all this way to look at what? And you know, Jody Rowley, who developed Frog ID, has developed a following of tens of thousands of people all over this continent who are super into this, and there's now articles in the media all the time. And it's kind of changing the positionality of frogs in Australia, and there are going to be more people who are starting off with the app, but getting beyond it. So I tend to be optimistic about the rise of citizen/community science. That's one of the bright spots in this whole constellation of stuff that we're talking about, where, quite honestly, there's more people now who are into getting out and IDing things and being engaged than there were before these apps were around. So I don't know, I'm interested what other people think, but I think we should embrace it and use it as a foundation for where you can go from there.
Hi, I'm Inbar Mayan, NSF postdoc at Harvard and Rutgers. I have a question about stewardship. And I think for all three of you, there's sort of ways in which you've probably interacted with keeping information or specimens. I thought what you described about keeping things in a hut in Malawi was a really cool thing. In terms of specimens that need special preservation conditions, what are your thoughts about the way that stewardship is done today and sort of what is the future of it? And with that, I mean sort of giving materials to collections that do have the facilities to preserve them, like cryogenic collections and the like, and sort of what the future of that might be as we move forward and think about more about where samples belong, but also how do we preserve them for the people who have sovereignty over them, and how do we preserve them for sort of the future of research in general?
I'll say a couple words and then turn it over to my colleagues here. But special conditions in all the meanings of that term have become really, really important in museum collections. The more we learn, you know, we've been doing things wrong or inadequately for a long time for some kinds of specimens. And just the way material comes into a collection through a freezer and all these protocols we have for what happens as they come in, how we take care of them, light exposure, you name it. It's all changed. And, you know, the art gallery at Yale installed cabinets that are filled up with nitrogen. And, you know, there's all kinds of things that are out there that people are trying out. And we're learning more and more about how to do right by these things as material objects and just the kind of physics and chemistry of it all. At the same time, there are all these considerations that are coming from source communities. So folks who, whether they're interested in repatriation or not, are interested in care. And some of that can be spiritual care. And so, you know, we have a flute that gets played, and we have, you know, there are objects where the source communities or makers are interested in certain things that we try to accommodate. We have to really think about the sustainability of all this, because it gets really complicated when you have 14 million things. If everyone has to be a special flower, you're in trouble. However, the objects that we've already taken on, we have a responsibility to them, and there's so much, as you're describing, negotiation that's so particular to different indigenous communities and other source communities that we have to honor that. And all of us in the museum world, like a huge fraction of my time is around repatriation, dealing with, you know, interacting with source communities, figuring out the right disposition of objects. And, you know, what's interesting that doesn't get as much attention is, I would say as often as not, the disposition is to have it remain in the collection. And then, but there's always a but after that. But we would like blah, 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 blah. We want to be able to visit it. We want to leave offerings. We want to do this. We want to do that. And that is a big part of the future. And all of this, and some folks said this yesterday, this takes a lot of time. And what you're doing is you're developing relationships that build trust. Whether you're doing field work, whether you're dealing with a collection, everything that you all were talking about, it's relationship building and trust.
I'll add just a brief couple responses to your question. In part, I've answered what happens to many of the samples we collect, and we destroy them, and so they have no longer lasting legacy. In some cases, like with the Ancestry Project, we were given a permission to use the samples. We work with them, we analyze them, but we kept all the remnants of the bones, the bone powder, the dental calculus, anything that we used that was part of the ancestors, as we call them, were then placed in the boxes with the other remains, and then into the crypt and reburied. And this was at the insistence of the community that all parts that were not used are actually, you know, repatriated with the individuals from which they came. So I think that actually is a very good way, at least in terms of the ancient DNA work, to go forward. And I think other communities are trying to emulate that sort of process.
For our collections, we have a range of different requirements we need to try to meet, and sometimes they're contradictory. So for some groups, it's very important that no light strike an object. For other groups, it's required that a light be left on in collections at all times. And fortunately, we've been able to negotiate ways to accomplish that. Sometimes the problems of technical conservation conflict with traditional care. More often than not, it turns out not only do they not conflict, but over the course of my career, we're kind of moving toward traditional care because the conservators are finding it works better. That instead of encapsulating things in plastic that doesn't breathe, it's actually better to have it in organic materials that do breathe, because the thermohygrometers are happier that way. You don't get hygroscopic materials having the same degradation. So many of the things that I had understood to be contradictions early on are proving to work pretty well. But it's always a negotiation. I remember when I was working in Milwaukee, we had one part of the collection where we had to mark all of the containers because the things in them had to be oriented in a particular direction. And they're in a container, you can't tell which way they're oriented. So you have to put them back the right way and then mark the container so that it's pointing in the direction that it needs to be. And that was a wonderful learning experience because it took us a long time to understand both what they wanted us to do, but why was more important. And once we understood that, we could come up with all kinds of ways of accommodating that need. And it helped us a great deal. And by the way, I'm not even sure it is something they would have wanted, this particular community, but we were able to record all that because it made that object far more meaningful and gave us far more information about its context and function. So we benefited enormously from that interaction.
Paul Barber, UCLA. I want to raise an issue that I think is growing, and that is essentially the issue of orphan collections. So there's a lot of collecting that, historically was done by people working in museums. In the field that, I entered as a graduate student, students and faculty were going out creating their own collections to do genetic studies with. And, like at this point, I have a collection of probably 40,000 samples that have been, preserved for genetic analyses. They've all been done legally, have permits for everything. But, at some point, sooner rather than later, I'm gonna retire. And, this is it's a tremendously valuable collection, but it can't really go anywhere because, the museums don't want to take that on because, the landscape has changed a lot between when those samples were first collected and now, and, taking ownership of those, taking on the potential liability of having those, it's like no one wants to touch that. And so, given that we used to live in a world where most collections were done by people working in museums, and now I would suspect that most collections are not, like what's the ethics of going out and doing the kinds of collections that Scott was talking about yesterday, if at the end of the day, when the researcher retires, all of that's just going to be destroyed, and there actually is no long-term value in those collections. So how do we deal with this problem?
Well, I'll take a cut at it. We're not going to solve this in this room right now. We have a bunch of folks in the molecular biology realm in the medical school who are in exactly the situation you're talking about. Our provost just closed up his lab and had this brigade of -80 freezers filled with his entire career's work. And in general, what I can see happening is the students are like, well, I'll take some, you take some, and the stuff gets all fragmented. And meanwhile, at least on our campus, like right next door, you've got this institution that has professionalized doing exactly what they need. But to get to the point where you can have a smooth transition to moving that into a museum, the collecting needs to be done in concert with advising from that institution, and there needs to be some kind of an agreement. And we do have agreements with some non-curator faculty at Yale where we are planning to take their collections. So it does happen, but it's a small fraction. So it's something that we need to think about more. Not to pick on Scott's institution, but somebody sent me an article that was in the Crimson a couple days ago, and there's a human ancient DNA lab on campus, and their funding from a foundation's running out, and they're going, oh my God, what are we going to do with our samples? And you can't walk a block in Cambridge without running into a museum. They're everywhere. And so these institutions should be able to handle a database on ancient DNA, but that didn't show up in the article at all, so it doesn't even seem to occur to people. You can't just do it at the end, though. It has to be something that you're thinking about along the way, I guess I'd say.
I would add briefly that I've been involved in sample collection distribution to the protégés of previous researchers to be earlier in my career so that the work could continue on the same kinds of population history questions. I think there's now some concern about the, the extended lifespan of some of the samples. The Yanomamo case, for one, is and Xiangyang and some of the issues that arose there. They still echo today. One thing we also, is that going on, there are a lot of biobanks, especially in the medical context, for extended use for patient samples in various ways, and those being made available to other kinds of researchers looking at other kinds of diseases or conditions. I'm not sure that's a model that would be acceptable for anthropological genetics because of the issue of consent and extended consent by the communities who've donated those samples, plus the move away from actually establishing immortalized cell lines so that this DNA lives on forever beyond the lifespan of the individuals who are actually donated those samples and can't actually consent beyond the point of their lifespans to further use of those samples. So these are issues I think we're all trying to grapple with, especially as these collections grow and we all become more senior in our careers, and how we manage this vis-a-vis the communities we work with, but also potentially the countries where we've worked and the ethics communities there that we might be able to engage in, and somehow the repatriating remains in that way to those spaces.
Just very briefly, I'd also note that this is a problem across all the field sciences, not just with genetic material. Faculty members do active research in the field, they generate collections, they make no arrangements or limited arrangements for what's going to happen to those collections when they retire or pass away. And then after the fact, institutions are trying to make sense of collections. In some cases, what you end up getting are boxes of material with limited documentation associated with them because nobody thought the documentation was part of the collection. That's a matter of training.
Okay, we have time for one or two more questions. Oh, yeah, yes, you were. Are you doing first?
Just Steve Beisinger, UC Berkeley. Just sort of following up on this question of how to deal with samples. Museums are a great place, but I've found that unless specimens accompany the materials in some way, there's pretty limited interest in dumping a lot of digital files, dumping a lot of long-term data necessarily into the storage there, because they suffer their own challenges to maintain their own collections and keep those things going. So like I think the SAFACA database is probably a little bit of an exception, given that it was a past museum director who generated a lot of those data. And so I'm not quite sure. I'm also concerned, well, I love the idea of being able to leave samples in country of where they're coming from. There has to be some kind of financial model to keep that up, because I've seen this in Venezuela, where I've worked for over 40 years, materials that were collected there through American Museum of Natural History and stored there have wound up in disarray because there really was no financial model to maintain that. Whereas another museum there in country has an endowment and a much smaller mission related that allows those samples to be maintained. So I don't know as you said, David, I don't know that there's an answer to these questions that we're going to have in the room, but I see these as the elephant that's stalking us here.
No, go on.
Very quickly, I completely agree, Steve. I think that we are building our capacity to take on digital collections beyond former directors. So I would push back on that a little bit. I mean, this is something that we view as being really important. So I think that we'll see growth in that. The other thing that worries me a lot is a lot of our repositories that a lot of people are using, they get an NSF grant and they say, well, I'm going to dump everything into Dryad. The curation of what goes in there, somebody did a study, and less than half of what goes into some of these repositories is usable. And a lot of them are grant funded, and there isn't a long-term vision for them. So there's going to be a reckoning on all this. And so I don't know when we'll get smarter about this, but we do have to fund institutions in perpetuity if we want them to take care of things in perpetuity. There's just no real way around that. The other thing that's quite clear is what whether it's Malawi or in other parts of the world, there need to be more museums in more parts of the world. You know, the answer can't be that we just coagulate collections into the Smithsonian and the American Museum, as great as they are. They can't take all the world's orphan collections as institutions one by one, you know, say uncle and get out. That can't be the way this goes. Collections, for all the reasons you've been hearing about, are too important, and they, you know, everybody that does field work should be thinking about what happens to the data, the specimens, your collections. That should be part of your planning, and it needs to be different than it is now.
May I add one quick comment?
A quickie.
I just wanted to tack on to the end of that comment about data or collections, is at least with data repositories, there are those sorts of issues, but also what we're encountering is that communities, we ask them how they want the data to be preserved and publicly disseminated, and many are choosing different kinds of models for that. Some more restrictive, some not shared, at least to the extent possible. Others are more open to sharing it more broadly. So that too then is a basis on a population by population basis, and then makes visible different data in different forms depending on their community's preference for sharing.
Okay, the last word.
Thanks so much. My name is Julia Giblin. I'm from Quinnipiac University, and I wanted to ask you all that a lot of these talks today have talked about more ethical stewardship of collections, and at the same time, like the broader conversations we've been having still seem to be focused a little bit more on kind of primary research and that kind of Western approach to knowledge. So I'm really curious where you all see kind of the future of knowledge generation, especially as we have more community, indigenous-driven, you know, not only stewardship of collections that have been collected in the past, but like where do we go from here if we continue to generate new collections where kind of a more Western approach is not driving the questions? So I'm just curious your thoughts about that.
Well, I'll just say very quickly and then turn it over to folks who probably know more than me, is that we get requests to use our collections by all kinds of folks for all kinds of reasons. And it may be somebody who is a basket maker from New England, from an indigenous community, who views the basket collection at the Peabody as a reference set. And having a set of baskets from all over the region done by different people at different times is actually quite useful. And so it's not a traditional Western scientific use of the collections, but we have people doing all kinds of things with the collections, and our criteria are, you know, we don't want things to get damaged or destroyed, but and we have to think about bandwidth sometimes, but you know, within that, there's lots of room for lots of different kinds of activities.
We have about 7.5, 8 million objects in our collection, and we're in the midst of re-inventorying it all. Part of the reason for going with notices and labels is we're trying to work toward an informatics model that we have an informatics model we use to organize collections for our purposes, but to have other facets to that informatics model that meet the needs of the community and are organized in very different ways. And part of that also becomes a matter as simple as naming. Naming of sites, naming of ceramic types, naming of other things, because that taxonomy automatically creates groupings. And they're not the same groupings that the Native communities we work with would use. And that requires us to have models for organizing information that don't just serve our purposes, but serve other ones. And in some cases, they're ones that we have trouble understanding. It's not as simple as they give us a different model and we can immediately implement it. So there are challenges to it. But if I'm imagining what's going to happen 10 years, 20 years from now, collections are going to be designed, or collections databases will be designed so that there are multiple facets that allow you to get at information using different constructs depending on the kinds of questions being asked. I wish I knew how to do that right now, but.
I'll add briefly that many of the communities that we're working with are already engaged with museums and sometimes developing their own heritage materials within the communities themselves. They're utilizing various resources to explore history and culture and ancestry, and genetics becomes another tool to do that. So that actually facilitates conversation about the work we're doing and allows us to build or help to elaborate some initiatives that the tribes are already using, or various communities, some of which are also educational initiatives for the kids and the members of their communities. So I think and this is sort of a mutually agreed upon approach. So I think that many communities that we've worked with are not averse to more Western ways of sample collection and analysis. It's more they want to know how they're going to benefit from it. Why should they be involved with it? What do they gain from it beyond what they already know? What kind of enhancements will come from this kind of engagement with a very scientific or hard science type of an approach? And it's a mutually enriching process to learn from their perspective what they think the history shows and what genetics shows, and then figure out how we explain things that don't make sense together.
Okay, well, I want to thank the speakers and the audience for a very stimulating morning.